Hello, my name is Sabrina Templo. This is week 5 for Bio 225. Today is June 11, 2020, and this lab experiment is going to be biochemical testing for microbial identification and carbohydrate fermentation testing. In this lab, I will incubate S. cerevisiae and S. epidermidis and glucose, fructose, and mannitol to categorize metabolic properties. As you could see, I can already label all my phenol red. And then number 1 is glucose. 1 is going to be with S. cerevisiae, and the second one is going to be epidermidis. And then number 2 vials is right here. I already completed these ones as well as the third tubes. The second ones is the fructose. And the third one is going to be mannitol. So I'm going to show you how to do this with the glucose. So first what I'm going to do is take my phenol red and I'm going to put it over the flame. I'm going to use the S. cerevisiae. Then I'm going to get my glucose powder, and I'm only going to put half of this powder into this in the phenol red broth. So I'm going to put half of it in. Okay, and now I'm going to sterilize the lid and then recap. And then I'm going to shake the broth until the powder is dissolved. And then I have my Durham tubes, which I've already soaked in alcohol so that they're sterilized. And how I'm going to insert this into the broth is the tube opening of the Durham tube has to be open towards the bottom of the vial. So the opening of the Durham tube is going to be downwards. So I'm going to open and sterilize. Now I'm going to insert the Durham tube with the opening in the bottom. And then reflame the lid and then close it. So I have to make sure that there's no air bubbles, so you invert it. And then flip it over and it's in the bottom now. Now I'm gonna get my S. cerevisiae broth, a culture broth. So the next step is I'm going to sterilize the lid of my culture. And then I'm gonna sterilize the lip of my phenol red, S. cerevisiae. Now I'm going to get my pipette for my epidermidis, and throughout the procedure I marked it as E and C so I know which pipette is for which bacteria. Now I'm going to make sure that is dry. And then I'm going to collect 4 drops from my S. cerevisiae vial. And then I'm going to put it into the phenol red, 4 drops. And then this is going to go into the bleach because I'm already done with the other ones. So I'm gonna reflame, and then recap the culture broth. And then reflame. And then for my culture broths, I'm gonna put it back in the refrigerators for future experiments. And then I'm gonna put these phenol reds in the incubation area for 12 hours. And after the 12 hours, I will observe them for carbohydrates fermentation results. And if there's no changes after the incubation, I will check it again for 6 hours. After that, I will observe with each broth, noting the color of each solution and the presence of gas bubbles in the germ tubes. The fermenter with acid production will be in the color of orange. The fermenter with acid and gas production will be yellow, and no fermentation at all will be no color change and will remain red. So that concludes my experiment for carbohydrate fermentation. Thank you.